Hi everyone. This video is to walk you through the Polymer Lab. We're going to jump back to some biochemistry topics and talk through a little bit about how we make these big macromolecules. Remember, we have 3 macromolecules that can become polymers: nucleic acids, carbohydrates, and proteins. Lipids do not make polymers because they do not have a monomer or a repeating subunit. So keep that in mind as you go through. This chapter has some really good review attached to it, or I should say this lab has really good review attached to it. So this might be a nice one to come back and study from before exams. Make sure to read through the entire lab procedure. Our experiment today is a pretty fun one, so hopefully you guys enjoy it. And I'm starting with part A. So part A says making a polymer. Poly means many. A polymer is a long chain of more than 5 repeating subunits or smaller molecules. Your carbohydrate chapter says that it can have more than 9 or 10 when we talk about the difference between oligosaccharides and polysaccharides. So slight distinction there. But typically a polymer needs at least 5 monomers or more to be considered a polymer. Monomer, of course, means 1, hence the term monomer. A dimer means 2, di being the root for 2. So it asks you here, explain the relationship between monomers and polymers. So put that in your own words. If you had 2 separate monomers and you wanted to combine them to make a dimer, how would you link them together? So think through our functional group section. What functional group allows us to link 2 things together? Okay, now it's kind of showing you pretty clearly. All you need to do is just look at those 2 monomers, see the functional groups, and remember how we link them together. Maybe your making molecular train section will be good to help you out with this if you forget exactly how to do it. Number 3, you had to remove something. What did you remove? And if you combined those 2, what did you get? So you removed 2 separate groups. Okay, to answer number 3, you removed 2 separate types of atoms or molecules. If you put those 2 things together, what would it be? What would it make? All right. Now this brings us into the bottom of the page. It says, think about what it is called when you remove water. When you combine monomers into larger molecules, a water molecule is removed. Remember, this is dehydration synthesis. We're synthesizing or building by pulling water out. So there's your answers. We remove water to make something new. Is this process new to us? No. We've seen this a few times now, right? So again, you can look back at your making molecular train, or you can look at your functional group section to read through a little bit about how this all works if you forget. Number 5, this is a process that is invertible. It can be undone. How would you take a dimer and make it into 2 monomers? So how would you split it apart? Do the reverse. Okay, go ahead and write that out. And you had to add something. To do the opposite, you had to add the same thing that you pulled out. So number 6, what did you have to add? And then it says down below, adding water back can split the dimer. But you must also split the water into the separate H and OH, right? We can't just leave it as H2O. This is called hydrolysis. Hydro meaning water, lysis meaning split. And we just made a dimer, which is only 2 monomers. It's not a polymer yet. So now we're actually going to look at making a real polymer. Okay, this is a pretty fun lab to do. Maybe you try it again later with some kids. Again, please be safe about it. Okay, but it's a really fun lab for the family to get involved with if you feel like everyone can be safe about it. You're going to be using a couple chemicals today, so please make sure that you look up the SDS safety information online, like we showed you in the first lab. Go through, read everything just in case anything ends up getting in eyes or whatnot. Hopefully it won't. Be careful. Okay. We are going to be making a polymer from some Elmer's glue, some borax, which is essentially like a liquid detergent. Okay. And then, of course, some water. You might also want your food coloring if you want to color it a pretty color. And you're going to need a few other things too. You'll need your scale, a plastic spoon. You'll maybe want your filter paper to help you weigh things out on, so those coffee filters. All right. You're going to need your beakers that were included in your lab kit as well. And of course, grab your safety goggles. I'm going to go to my lab and we'll get started. Okay, so once you are in your lab, make sure you have everything set up and ready to go. Hair is pulled back if you have long hair. Any earrings are out of the way. Make sure you have your safety goggles with you. And we're going to start out with weighing some of our borax. So it says place a piece of the filter paper onto the balance. Make sure that if you do that, you zero it. Now you can also weigh directly into the plate. Just be sure to wash it afterwards if you do that. You can also bend this a little bit. It's a bit hard to see otherwise. Okay, so I'm at 0 right now. I want 4 grams of my borax. And that's a little too much. So if you have too much, you can take some back out, no problem. And try to be as exact as you can be. Okay, I'm at 4.00 grams. And now it says, use your graduated cylinder to measure 100 milliliters of water. Tap water is fine. So hopefully you have your 2 beakers with you, okay? So I have 100 milliliters of water. You can also measure this out in your test tube and add it to this, which is the more accurate way of doing it. And then you want to have the 4 grams of your borax and put it into your water and stir well. So this is where your plastic spoon will be helpful. Make sure to shake off all of it. And then go ahead and stir. You want to stir from the bottom. Make sure you get it to dissolve nicely. It'll be a little bit cloudy. All right, and next we're going to be using our different beaker. Now I chose to use a smaller beaker for the borax solution, and I'm going to be using my bigger beaker for the glue. So if you do have 2 different sizes, I suggest doing it that way. In our larger beaker, I'm going to add 75 milliliters of Elmer glue, which should have been in your lab kit. Might actually be faster just to pour it in. Here we go. Come down so you can see. It's going to end up being a pretty decent amount of the bottle. Still need a little bit more. Okay, so maybe you pour it in like me, let it settle for a minute. And if you need a little bit more, you can squeeze it in with the lid on. Actually, right around 75. That's pretty good. Okay, now in the exact same beaker as your glue, you're going to add 75 milliliters of water on top of it. Just regular tap water, not the water that you've mixed. Okay, so 75 milliliters into this. So you want your total marking to be up. Here we go. And you should be at the 150 mark. So you have your nice 2 layers there, your water and your glue. Okay. All right, now that we've done that, we want to add a few drops of food coloring if you would like. That's always fun to do. So go ahead and add your food coloring in now, and then you want to stir it for 1 minute. Okay. Now if you use your borax spoon, just rinse it off before you put it in the other beaker. Now you have your glue, your water, and maybe your food coloring. Go ahead and stir. And it's going to take a little while to stir. You'll notice it's pretty clumpy. We want to try to get it nice and uniform. And so that's why I like the bigger beaker. You have a little more room to work with. And we're also going to be adding a little bit more to this as well. Now after you've stirred it wants observations. So observe your glue and water solution. Write down some information about it. What's the consistency like? What's the color like? Is it liquid? Is it solid? Is it sticky? Is it runny? Whatever you see. And while you do that, I'm going to keep on stirring here. Okay, so I've now stirred up my glue. It looks nice and consistent. I added a little bit of yellow food coloring to mine. It's pretty light. And so now that you have it nice and stirred, you've made your observations, you're going to go ahead and combine your two solutions. So you want to take your borax water and pour it in to your glue water in its entirety. Okay. I had a little bit of borax left over there. That didn't quite dissolve. And now you want to start stirring. So stir from the bottom. Make sure you get everything mixed, and you're going to stir until it's too thick to continue stirring. And you'll see it's starting to thicken up on my end. How well you can see that. Getting kind of a glob in the middle. You can see it's getting pretty thick there. But keep going until you get it very well mixed and you can't stir it anymore. Okay, so once you can't stir anymore, you're going to take your object, for lack of a better word, out of your beaker. All right, paper towel is going to be nice and helpful because you'll notice it's a little slimy. And observe it. Okay, you've read the SDS sheets on all of this material, so you know you can touch it. Make sure you wash your hands afterwards. Play around with it. See, does it stretch? Maybe you roll into a ball. Does it bounce? Can you break it apart? Kind of experiment for a little bit. Okay. Have some fun with it. It's kind of a weird thing to touch, gotta say. Sticky, right? Sticking to my paper towel. So any kind of qualitative data you collect, observations, write it down. Right, maybe the more you manipulate it, the less liquidy or sticky it becomes. Kind of a strange thing. I really can't describe what it feels like. You'll have to try it out for yourself. Okay, so once you have your observations, maybe you play around with them a little bit more, and then you are finished with your polymer. So this is our nice big polymer, okay? Write down all of your qualitative data that you found. Any kind of interesting information about this oozy kind of blob, what you think about it, and think about how it's different from the qualities it started out with. So how is this different from our liquid glue? How is it different from our glue and water solution, our borax and water solution, okay? And how did we combine those liquid things to get something like this? Okay, now your procedure says the properties of the new material resulted from the glue molecules being linked together by the borax molecules. This is called polymer cross-linking. So your glue, your Elmer's glue, had long polymer chains in there already, and the borax was able to connect them, to string them all together, to combine them to make a larger polymer, okay? Think about it. What would have happened if too little borax were used? So if you didn't use all 4 grams, say you only put in 1 gram, what do you think that solution would have looked like at the end? And then it says, does the new material have some characteristics of a polymer? So think about polymers. Think about the polymers we've talked about. This might be a good place to research polymers, okay? Polymers exist all around us. Mostly synthetic, certainly, but they exist in our daily life. We use them for just about anything. Maybe your shirt has polyester in it. Maybe you use polyurethane in your tubing in your house, okay? All sorts of different polymers exist in our plastics, in the containers we use for our food, in our sheets, in our clothes. And again, a lot of them are synthetic. We have a lot of different synthetic polymers that scientists have created to make life easier for us, okay? But there are also the natural polymers, things that we've talked about in this class in terms of the body, and polymers that exist out in the wild, like cellulose, for example. So they're all around us. So maybe you do a little bit of research and you find some specific characteristics for polymers. Does your new material, that end result, did it have any characteristics of a polymer? If so, what do you think? What were they? And then it says you can dispose of your polymer in the garbage, so it's safe to just toss that. You want to rinse out your beakers. Make sure you flush the drain with a lot of water, especially if you have residue left over. Wash your hands, wash your station up, and then make sure to save your beakers and anything else for future labs. We're going to move on to part B. So part B is a nice review of the 4 macromolecules that we've talked about. It says biochemical compounds are organic compounds that can be made by living things. There are 4 large molecules or macromolecules that are essential to life. Some of them are polymers, some are not, which we've discussed. The 4 are carbs, proteins, nucleic acids, and lipids. So we're going to start out with carbohydrates. Carbohydrates are, of course, the main energy source for living things, right? That glucose is very easy to convert into ATP, which is our cellular energy source. Carbohydrates are composed of carbon, hydrogen, and oxygen in a 1:2:1 ratio. And sugars are carbohydrates, but not all carbohydrates are sugar. So for example, cellulose, that big polymer building block for plants, that's not a sugar, but it is made up of a bunch of glucose or sugar molecules linked together. But when you eat cellulose, think of eating like a really fibrous broccoli stalk. Okay, that doesn't taste sweet, it doesn't taste sugary. So not all carbohydrates are sugar. Some certainly are, though. Glucose is a simple sugar, right? It's a monosaccharide, a monomer. Glucose comes from the Greek word glucos, meaning sweet. And of course, that OSE ending tells us that it's a sugar, just like maltose or lactose or fructose. Right? Glucose is the energy-storing molecule formed by plants through photosynthesis, right? That anabolic chemical process. Glucose is then broken down by both plants and all the organisms that eat plants through cellular respiration. That releases the energy that's stored in glucose so that we can make ATP. And we have to make ATP, we can't just use glucose, because the cells will only accept ATP. Okay, so even though plants are using photosynthesis to make glucose, they're not making it for everyone else. They're actually making that glucose for themselves too. So plants do use cellular respiration to break apart that glucose that they've built. And remember, they store it in terms of starch. So they store their extra glucose all packed together in a big polymer called starch. And as we've talked about, animals can't do that because we have to walk around, right? So that's why we convert it to fat instead, if we have excess energy. Okay, can you think of some other sugars? All right, I've named some already. Either look in your book, do a little research online. What are some other sugars that you can think of? And if you think of them, where are they found? So where do we find lactose? What is fructose found in? It says glucose can come in several different forms. Okay, the molecule itself can be shaped in a couple different ways. It can either be in a chain, which we see on the left, or it can actually twist into a ring. So just think of taking that chain and connecting the ends. If it's in a chain formation, that oxygen is really important in helping it bond. Does everyone see that in your molecule structure there? So we're actually taking that oxygen and we're helping it bond with the carbons. The model on the right assumes that you know that there is a carbon atom wherever 2 or more lines meet, and there's no other letters there, right? We know that by now. Carbon and hydrogen can just be represented by those mountains and valleys, right? Okay, what is the molecular formula for each of those? Take a look, count up your molecules. These are both glucose molecules, but they're in different forms there. Now, it says these molecules are going to have the exact same molecular formula, okay, but different structures. So that makes them isomers. Isomer is your vocab word for today. Iso means the same or equal. So an isomer is the same molecule, just oriented in a different way. Scrolling down, it says a disaccharide is when 2 simple sugars are bonded together. Sucrose is a glucose molecule and a fructose molecule bonded together. Sucrose is our table sugar that we typically use. So sucrose is actually a dimer, right? It's a disaccharide. So is a disaccharide a polymer? What do you think? Think about the definitions. Is a disaccharide a polymer? Carbohydrates can form polymers. When two monosaccharide molecules are bonded together, a water molecule leaves the molecule. Of course, we saw this up above. What was that process called? Number 4. A large amount of glucose molecules, anywhere from 3 to 1,000 of them, can be bonded together. This can help form starch, okay? What is the polymer in this example? So that above sentence, which one is the polymer? And in that above sentence, which one is the monomer, okay? So a large amount of glucose molecules bonded together form starch. Which one's the polymer? Which one is the monomer? And it says athletes often consume mass quantities of these complex carbs before competitions. The human body begins to break down that starch into glucose, and then the glucose that isn't needed right away can actually be stored as glycogen. Glycogen, if you remember, is a polymer, how we store energy in animals, right? We usually store it in our muscles. So remembering what is formed when two monosaccharide molecules bond together, what must be necessary to break the monosaccharide molecules apart? So again, this is thinking about the beginning of this worksheet. When we break something apart, what do? What do we take out? And what is it called? Now we're going to jump into proteins. It says proteins are polymers made from amino acids, and they have many structural and functional purposes, right? Proteins are so incredibly important for the body to function. Really for any life to function. Protein means of the first importance, right? They're vital for life. They are much more complex than starch. The basic unit of a protein is an amino acid, and remember there are 20 to 21 amino acids that occur naturally in nature. Amino acids are composed of carbon, hydrogen, oxygen, and nitrogen, along with some other things sometimes, like maybe sulfur. What is the monomer of proteins? The type and number of amino acids determines how the protein behaves, right? It also determines what protein it is. It's all about the order of the amino acids that will then dictate the shape that it takes and the function that it has. Now we eat protein, right? We break them down into individual amino acids through a process called catabolism, right? Catabolic reactions are reactions where we break apart bigger molecules. And then we rebuild them through anabolic reactions or synthesizing reactions to form the proteins that we need. So even if I eat protein in one form, my body can take it apart and build it into another form that might be more useful for me. Some amino acids are able to be synthesized by the body, while others have to be ingested. The ones that we need to eat in our diet are called essential amino acids. Essential because our body cannot build them. Okay, so if you remember, when we talked through proteins in our lecture, I said not all proteins are created equally, right? Some of them we have to eat. And if we don't eat them, our body's going to start breaking down because we can't build the proteins that we need. We won't have the right amino acids. Now, this all goes back to eating a nice balanced protein diet. So those of you who eat animal meats or animal products like milk and eggs, you're getting complete proteins. You're getting proteins that have all of the essential amino acids. If you're a plant eater, a vegetarian like me, you have to be very careful on eating a nice balance to make sure that you get both your essential amino acids mixed in all the different proportions, right? If I just eat beans, I'm not going to be getting all of my essential amino acids. So I need to make sure that I pair it with other types of protein sources. Animals, though, because they need to use all of the amino acids, they have all of them ready for you when you eat it. Now, nucleic acids. It says these are the polymers that store our genetic information, right? Our DNA and our RNA. The monomer for a nucleic acid is a nucleotide, which is made up of a nitrogenous base, a sugar, and a phosphate group. They are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And remember, ATP is a nucleotide, right? It is that monomer. DNA is a nucleic acid with the shape of a twisted ladder or a double helix, while RNA is only single-stranded. Most of your cells have a copy of your DNA, almost all of them, okay? And it contains a code or a blueprint for how the proteins are going to be made or synthesized in the cells. The sequence of the monomers in DNA determines the protein that is made by that cell, okay? So this is all about the central dogma. The central dogma says we have a whole bunch of DNA, a whole bunch of instructions. That DNA is going to be transcribed. Think about writing, right? Transcription. It's going to be transcribed into RNA, which is a smaller molecule, and it's able to leave the nucleus. That RNA is going to go out into the cell, and it's going to be translated into an amino acid sequence. So DNA to RNA to amino acids. That's how we make a protein. So the DNA is really storing the genetic information, right? It's telling the body what proteins to synthesize, which then tells the body how we're going to actually express all of our different traits and our genes. What is the monomer for nucleic acid? And what are the two types of nucleic acids? Nice and simple, write those down. Now, of course, you want to remember that in the central dogma, of course, it's kind of central to the whole theme of life, how things are created, how we pass along our DNA. And always remember that there are mutations or errors that can happen as we're translating and transcribing. So the body has different ways to protect against that. So maybe go back and proofread checkpoints that are going to stop and make sure DNA was copied correctly. But it's quite a big process. It's a big process to pass this DNA on to every new cell, and also to translate it and transcribe it to make our proteins. Now, our last macromolecule is lipid. Lipids do not form polymers, but they're still very important, right? They help with a whole bunch of different things like signaling, insulation, protection, storing energy. They're a major component of biological membranes. Think about our cellular membranes. And then, of course, we also have our triglycerides. Now we're down to the analysis, okay? Talk about why people eat carbohydrates, all right? Why is it good for us to eat carbohydrates? Now, there are some people who use keto diets where they have a very low level of carbs that they're eating, very minimal, okay? And that is possible to sustain. I wouldn't say it's necessarily the healthiest. And think about that. If you don't eat any carbohydrates, what would happen? Can you keep that up for an entire life? Why would it be necessary to eat carbs? Or is it not necessary to eat them at all? What do you think? Number 2, you want to compare a saturated and an unsaturated hydrocarbon chain. You want to use 3 carbon atoms, so draw this out. Remember, saturated refers to the number of hydrogen atoms. So if it's saturated, it is an alkane. If it's unsaturated, it's either an alkene or an alkyne. Okay? And remember that every single carbon that you draw can only have 4 lines coming off of it. That's it. It needs to have 4, but no more. Draw your chemical structure and then write down the molecular formula. So using 3 carbons, making 1 saturated and 1 unsaturated. Draw it out, write down the formula. Some polymers have a memory, meaning they return to their original shape. Okay, this is like, if anyone has ever bought a dress shirt that is wrinkle-free, meaning you don't have to iron it, you can crumple it and it won't wrinkle. That's a polymer that has a memory, we say. So it will return to that nice, crisp, smooth texture, okay? Another cool example of how we use polymers in our everyday life without even realizing it. So not that example, but give me another example of a polymer with a memory. Maybe you do a little bit of research if you don't know off the top of your head. And then give me an example of a polymer that does not have a memory, okay? Human-made polymers are usually plastics, sometimes fibers. Do a little bit of research about these. see all of the different types of synthetic plastics and fibers that we use in our daily life. Okay. Now, a lot of you know this can become a big issue in terms of conservation, right? These plastics, they don't break down for thousands and thousands of years. And so when we throw them away regularly, single-use plastics, right, they start to build up and we have this big problem of overflowing garbage, nowhere for it to go. It ends up in our ocean, all sorts of sad things. This happens too when we have synthetic fibers in our clothes. So when you wash your synthetic clothes, some of those little plastic fibers, you won't be able to see them, but they'll actually get washed away into the waterways and into the ocean as well. So think about all of the different polymers that we use that really are quite great for helping us be comfortable and having a more efficient lifestyle. But think about the flip side of it. Okay, there are some downsides, especially to the wider world. What might those be? So find and describe one polymer that's a plastic, and then find and describe one polymer that's a fiber. Give me a little bit of information about it. What did you find out? Number 7, you just need to complete the table. So is it a polymer, yes or no? And if you said yes, what is its monomer? Number 8, in your own words, describe the main functions of the 4 macromolecules. Why are these important? What do they do? And that's fine just to put in your own words. And then number 9, you're going to do a little bit more research. So you're actually going to research amino acids. We have 20 to 21 amino acids depending on the source, who you ask, okay? Categorize them. Write down the ones that are non-essential, and then write down the ones that are essential. Make sure you spell them correctly. This will be a really good place to study from. I expect you to know the essential amino acids, okay? You don't need to know all of them for this class. You might need to know that for a future course, but for this purpose, I want you to know all of the essential amino acids. So make sure you write them down, you do your research, make sure you spell things correctly, and then this will be a very nice study tool for you later on. Okay, that brings us to the end of our lab. As always, I hope you have fun with it and reach out if you have any questions.